And welcome to today's Chardan leadership call featuring Renovacor, a company advancing gene therapies for indications driven by BAG3 dysfunction. And a special thank you to our guests today, Dr. Magdalene Cook, President and CEO, Dr. Marc Semigran, CMO, and Dr. Matt Killeen, CSO of Renovacor. And I'm Geulah Livshits, a biotech analyst at Chardan, where our focus is on identifying companies that can generate exceptionally high long-term investment returns by creating and sharing in value for society. During this call, we will not be discussing any Chardan research. Any discussions about Chardan research should be coordinated between participants and their respective salesperson at Chardan. The recording of this call will be made available to clients after the call, and our compliance team has asked me to read this statement. By participating in this call, our speakers attest that they have made Chardan aware of any potential conflicts, and they will not discuss any material, non-public, or confidential information that they're aware of that may breach their legal, regulatory, or fiduciary responsibilities to any parties. And in terms of format, this is a 45-minute to 1 hour long webinar. Participants can ask a question by entering it into the Q&A box in the Zoom window. So with that, let's get started. Thank you all for joining us today. And to start off, Maggie, would you give us a high-level overview of Renovacor as a company and its history for those who might be new to the story?
Sure. Thank you, Geulah, and thank you so much for arranging this call for us. We're very happy to be here to share the story with your audience. So at Renovacor, we're bringing the promise of precision medicine to cardiovascular disease. In the past, for decades, we've treated cardiovascular disease with really one-size-fits-all approaches targeted at very large populations. And it's our view that we're on the cusp of a major transformation in cardiovascular disease to a more targeted approach that really gets at the root cause of pathology, much like we've seen in oncology over the past decade, where targeted approaches have really changed the way we think about cancer patients and how to treat them. So we're excited about that transformation in cardiovascular disease and being part of the early stages of that alongside peer companies like Rocket, Tenaya, MyoKardia, which was really one of the first to approach that. Our lead program is really the exemplar of what we're trying to do. So we are targeting a patient population where disease develops due to a genetic mutation. And we know what the cause of disease is in this group. It's a haploinsufficiency or a deficiency in the BAG3 gene. So we've developed a targeted approach, a gene therapy that replaces that missing gene, restoring cardiac function and preventing progression of disease. That's kind of the paradigm of what we're hoping to do with the getting at the root cause. Then we would look to translate what we learn in these smaller populations to larger, more prevalent groups where the same mechanistic drivers are at play. So moving from that rare disease group to the more common, larger group where it's the same pathology. And that R&D paradigm that we're taking forward with BAG3 is really one that we would replicate with other genetic drivers or targeted approaches to specific cardiomyopathies. So that's what we're really all about, and we're very excited to bring this lead program forward as an example of how we can apply this R&D paradigm.
Fantastic. So before we talk about the program and get into the science in more detail, can you take us through the key catalysts that are anticipated in the near term and over, say, the next 12 to 18 months?
Yes, very exciting time for Renovacor as we move into the clinic. So major milestone coming up, of course, is the filing of our IND for our lead program, which we expect in mid-2022, and the launch of our clinical trial, phase 1/2 clinical trial in our patient population shortly thereafter. As we move toward our IND filing, we have a number of studies underway in our haploinsufficient mouse model, so in our disease model, as well as in our large animal model, that will be generating data that we will look forward to discussing in advance of that IND filing. And then as our pipeline progresses, we'll be very excited to talk about which programs will be prioritized and what the timeline will be for those for moving them into development as well.
Great. So let's get into the lead program a little bit. So the lead asset, REN001, is in development for dilated cardiomyopathy with BAG3 mutations. So can you start off by giving us a little bit of an overview of this indication with respect to the market size, the current treatments, and the unmet need in this type of patient population?
Sure. Okay.
No, go ahead. I was going to say, Mark, that sounds like it's right up your alley.
Thanks. And Geulah, thanks for inviting us. And it's a pleasure to tell the Renovacor story to the group. So BAG3-associated dilated cardiomyopathy represents a segment of the very large heart failure population. And this segment of the population has dilated cardiomyopathy due to a mutation in the BAG3 gene. And, I hopefully will get to talk a little bit more about that gene and the protein in which it encodes. However, the patient population is relatively young. Mean age of onset is about 38 years of age. It is a familial dilated cardiomyopathy, and indeed, mutations in BAG3 are the third most common known genetic causes of dilated cardiomyopathy. It has a particularly aggressive course, with approximately 20% of patients having a major adverse event, a need for a heart transplant, a heart failure, death, a need for mechanical support, such as an LVAD, within 1 year of diagnosis. Patients are very frequently symptomatic when they're first diagnosed, and about 20% of them severely symptomatic with class 4 heart failure. So we have a segment of the heart failure population that, again, there's a familial component, a genetic component that's readily identifiable, that's more aggressive than the average heart failure patient, and that attacks patients at a younger age. And if I could add one more thing, that the familial nature of this disease is, particularly devastating to patients and their families, because not only does the patient worry about having the disease, they worry about their siblings, their potential children or children, that if they're carrying this mutation, that it's going to be highly likely that they develop this devastating disease as well. It's what drives me to really seek a cure for this disease through a technology such as gene therapy.
Got it. That makes sense. And so you mentioned, the heart transplants and mechanical support. Do you have any additional color on the type of outcomes that patients currently have with the available care, and kind of where you hope to improve on that?
Right. So, I mean, the figures that I mentioned, that 20% within 1 year, for example, that's on the current standard of care, our neurohormonal inhibitors, our, beta blockers, ARNIs, etc. Heart transplant, I did that for 3 decades when I was at Mass General. It's an incredible therapy. However, because of the need for immune suppression, the problems related to rejection, you're really trading one set of problems for another. So the ability to use a precision medicine approach, such as gene therapy, to address the underlying cause and mechanism of disease is very exciting.
Got it. So in that same vein, in terms of disease biology, just wanted to get at what makes this indication particularly amenable to treatment with gene therapy from your perspective.
Sure. Well, we're focusing on the patients that have what we call truncation mutations or deletions in the gene, which is in fact over 80% of the mutations that have been identified in the BAG3 gene. They lead to what we call haploinsufficiency, or decreased amount of BAG3 protein in the cell, particularly in the cardiomyocyte. And because of that, replacing the allele, remember, we all have two sets of genes, and in these patients, one is usually normal or wild type, and the other is affected by the mutation. So replacing that mutated gene, which is making this functional protein, if it's making functional at all, with another copy of the wild type gene through a technique such as viral mediated gene therapy, we really think that we can have a significant impact on the disease with a single episode of treatment because of the durability of the effect, as well as really approaching that underlying mechanism and being able to restore the gene function, cardiac function, and to decrease the number of these other medications that patients will have to take.
Yes, and also I think a key aspect here as well is the pleiotropic nature of the BAG3 function in the cardiomyocyte. And maybe Matt, you could walk us through a little bit of how essential BAG3 is in function and why we believe a gene therapy is the right approach versus a potential small molecule.
Sure, Maggie. And, I think it's important to know that BAG3 is a really highly expressed protein in the heart. And it has, as Maggie said, this multifunctional really repertoire of how it performs. So it plays a role in regulating contractility in response to exercise. It provides a degree of structural integrity and support. It really plays a central role in controlling protein quality control in the heart and exerts very important effects mediating against apoptosis. So it has this broad regulatory role across many different functions of the cardiomyocyte. So I always think when you step back and look at all these broad pleiotropic functions, it's not too surprising that anything that reduces the levels of BAG3 in the heart, an artificial event that you can induce in the lab, or a truncating mutation, as you heard from Marc in our BAG3 DCM patients, anything that lowers the level of BAG3 really can trigger the cause of disease biology, can trigger the onset of disease. And as Marc said, our goal with BAG3 DCM, our lead program, is to target patients with a certain type of mutation that have low levels of BAG3. And it's quite simply with our lead program is to boost and restore those levels of BAG3 expression in the heart to really have a meaningful impact on the disease biology.
Great. So on that note, can you maybe walk us through the design of your lead asset, and in particular touch on the features that you believe make it well suited to address the BAG3 DCM population?
Sure, that's a great question, Geulah. And I love describing this because it is such a clean and direct relationship here. And I think that's what we've seen in gene therapy in general the past few years. Success has come when there's a clear and direct relationship between the causative gene and the gene therapy that's being applied to it. So in our case, this patient population is a monogenic population. The disease is occurring due to the mutations described by Marc and Matt. We've selected vector selection, obviously very important in gene therapy, and we're going with the AAV9 vector. This has been shown, as you know, to be the most tropic to the cardiac tissue of the AAVs. So we've learned over the past few years in animal models and preclinical development that AAV9 is really the best choice for the heart. More recently, our peer company Rocket has also shown in the clinic that they have been able to have good results transducing the heart with AAV9. And so I think, that's really a great advance for our field in general to see that and progress in gene therapy in the heart. Unique to Renovacor is our method of delivery. So we've got the gene therapy, which is an AAV9 BAG3, and we are delivering our gene therapy through a local administration. So we are really trying to get to the target organ primarily, which is the heart. Our patient population suffers from a primarily cardiac disease. The symptoms are related to cardiac dysfunction. So we can really hone in on that organ when we deliver our gene therapy. And the advantage is a lower total vector dose per patient. So by delivering our therapy first to the target organ and having a first pass there, we are expecting our clinical dose to be a log to a log and a half lower than you might need if you were to infuse it intravenously. And this is really potentially a big advantage, safety advantage for our patients, to reduce that vector-related adverse events that can occur with AAV gene therapy. So the procedure is nothing new, nothing novel about the procedure in itself. It's commonly done by cardiac electrophysiologists. This is how they place pacemaker leads. The catheter also is commonly used. So this is a intravenous, it's an intracoronary infusion. The catheter is placed in the coronary sinus in the venous side of the heart. We use a balloon, slow the flow. We're able to bathe the heart muscle in the gene therapy before it circulates systemically. And this is really where that advantage comes into play, where you have your first pass through the cardiac tissue before the therapy circulates. So those are really the three key elements. It's a monogenic disease, so we're directly replacing that missing gene, using AAV9, which we know targets the heart, and then really local administration to bathe the target tissue and use a lower total vector dose.
Great. So maybe just on the topic of the delivery approach and the administration, you mentioned that this is the same type of strategy used to place pacemaker leads. Can you give some color on how accessible this would be to the typical patient with BAG3 DCM and or other potential broader indications in terms of the logistics and the complexity of the procedure and the types of patients that might be addressable here?
I'll come off mute there. I'm sorry. Maybe I'll take that one because I've treated these patients. So again, this is a low pressure system, the venous system, and when, for example, electrophysiologists do this, they're rather rapidly able to cannulate the coronary sinus and the coronary veins. So I suspect this would be a fairly brief procedure that would certainly not require general anesthesia, for example, be done under a local. And, it is ordinarily done as an outpatient procedure where, there's just a few hours of observation after, say, a 1 hour procedure. And then, we in our initial investigations are going to continue to observe the patients because we want to be very conservative in terms of identifying any safety signals. On the other hand, eventually, this may well become a very easily tolerated procedure by patients.
So in terms of the locations that this might be offered, do you think this would need to be done at specialized centers or community settings, or kind of what's the accessibility of this type of therapy?
I think that as Maggie pointed out, this is done routinely as part of, pacemaker implantation by cardiac electrophysiologists, and that's fairly widely dispersed in our country, in Europe, and really in the rest of the world. So this is, it's a specialized procedure, yes, but it's, and even in what you might call secondary and tertiary hospitals, it is done here in the U.S.
And for those who might be familiar with other local delivery approaches within the heart, I've heard of Antigrade Delivery. Can you explain how this is different from that which has been tried in the past for a gene therapy approach in the heart disease space that has not been successful?
Certainly happy to. So antigrade delivery, through the coronary artery, usually that's a high flow system that you cannot occlude for a sustained period of time. As you may know, we do occasionally occlude coronary arteries when we're treating an obstruction, a coronary atherosclerosis, and when somebody's having an angioplasty. But the duration of occlusion with our balloon and stent deploying catheter is on the order of maybe 2.5 minutes. And we will be, through the coronary venous introduction of the therapy, able to occlude coronary veins and maintain, I believe, near normal blood flow because we have clinical experience with this, and there are many veno-veno collaterals in the heart, maintain normal coronary blood flow for a duration of time that, may be on the order of, 15 to 30 minutes to allow, as Maggie mentioned, that dwell time and that first pass effect of the viral vector to occur. I would also say that, by working in the low pressure venous system, that this is intrinsically a safer procedure than working in the higher pressure arterial system. And hence, I think, coronary angioplasty, I think, has low complications. If anything, my hope is that this procedure will have even fewer, if any.
Great. And just last question on the procedure itself, are there any typical risks that would be associated with this procedure that you're aware of that you'd be looking out for?
I think very rarely, and I'm talking in the 1 in 1,000 or less range, there can be some injury to the coronary vein, and frankly, it usually heals itself up.
Great. So let's, in that case, turn to some of the preclinical data that you guys have generated on this program. Can you maybe walk us through the key data that has been generated on BAG3, as well as your lead program specifically in heart disease settings, for example, in mouse models and in vitro work? And then maybe afterwards you can put those together and talk about your more recent experiments in the larger animal models.
Sure, Geulah, happy to take this one. So the data that we've generated from our academic founder have really fallen into 2 buckets. The first one is centered around our lead program, our lead target indication, so BAG3-associated DCM. So our founding academic laboratory and scientists developed a powerful genetic animal model of our target disease that really recapitulates many different aspects of the types of syndrome we see in patients that Marc mentioned earlier on. And we've shown that in this mouse model, if we treat these mice with AAV9 BAG3, we can prevent the onset of cardiac dysfunction rather nicely, rather prominently. So certainly very excited about those data, the potential and the promise of those data. And we can get into this shortly. We've also done quite a bit of work around developing and optimizing, just as you heard from Maggie and Marc, this local route of administration. So that's the data package for our lead program. Outside of our lead program, the second major bolus of data is around the promise of extending our same BAG3 gene therapy approach to other more common, more prevalent heart failure cardiac indications. So I think that's what's intriguing for me, and it's really, really exciting to know that you can translate potentially the biology of BAG3 to multiple other disease indications, especially the heart. And again, we've shown, and this has been published by our academic founder, that you can take the same AAV9 BAG3 approach and take this to settings such as heart failure, so ischemic induced heart failure. We've shown, and we've published this, that treating mice that have had a prior heart attack and have entered into a form of heart failure, treating these mice with AAV9 BAG3 can very quickly, within a matter of weeks, almost completely restore cardiac function in these mice. This is just one of the indication expansion opportunities we're looking at very closely. There are several others. So I think that's the second major part of our data generation, has been in the promise of extending this BAG3 approach to these more common acquired heart conditions.
Great. That makes sense. And then, so coming back to the route of delivery and the retrograde infusion, you recently shared some data on that in a pig model showing cardiac transduction. So can you walk us through those experiments and also discuss kind of any head-to-head comparisons that you've done evaluating the local versus systemic routes of administration? I think we've gotten some investor questions on that front as well.
Sure, no, happy to. So we realized very early on the need to scale to a larger animal model. Using the pig, it's a commonly used model system in cardiovascular physiology, especially in studying novel heart failure therapeutics. So we're really following what's been done in the field previously. The pig is a very well-established model for this disease setting. as well as also for toxicology and safety pharmacology studies. It's becoming a very powerful, robust, well-used model. So we really leveraged that infrastructure, that model, to look at if we could deliver our gene therapy locally and achieve a successful transduction of the heart. So we performed a feasibility study to really test the hypothesis: can we deliver locally to this region of the heart at a low vector dose and sufficiently transduce the heart? And by transduce, we mean, can we sufficiently deliver enough copies of the viral gene to regions of the heart? So we performed a feasibility study in healthy mini pig setting. We delivered our lead program, REN-001, via the retrograde coronary sinus infusion route of administration. We observed these animals for a period of time. And then after this, we looked at tissue sections from the heart. We took multiple tissue sections from many different regions of the pig heart and looked for how much transduction we saw. And we saw, really pleased to show that at our medium and our high doses, which are both under 1E13 vector genomes per kilo, we saw transduction at and surpassing a VCN, vector copy number transduction threshold of 1. So we're clearly at our mid and high doses, not only achieving, but surpassing the transduction threshold that we set, which means that on average, at least each cardiomyocyte contains at least one copy of the functional BAG3 gene. So it's certainly really excited, really promising data we showed in this feasibility study, which really informed the design of our ongoing GLP toxicology study in the same animal model.
Great. So a couple of follow-up questions on that. In terms of the pig models, you talked about how this is a model that's well established in the heart disease space. What's known about how predictive they might be for something like AAV safety?
Yeah, great question, Geulah. I mean, the pig heart, many anatomical, physiological similarities to the human heart. So it checks a lot of boxes for us in studying this disease. It is being used. There are studies and papers written about this, the use of the pigs for safety pharmacology, toxicology studies. If you look at the prior cardiac gene therapy field, many, if not most of them, have used a similar pig model to study efficacy and also look for certain safety parameters too. So it's our view that the pig model, especially for cardiovascular and heart disease, is going to become an increasingly used model for efficacy as well as toxicology studies. And certainly with our ongoing GLP tox study, one that study wraps up, we certainly look forward to presenting data from that next year.
We've gotten some questions around data timing for these preclinical pig studies. Is there, when should we expect data from the pig studies that you're advancing to support the IND?
Sure. So we're anticipating our core IND-enabling preclinical, non-clinical studies to complete to enable our targeted IND filing. And so we certainly look forward to presenting data from all of our preclinical studies in due course in 2022 and onwards. Maggie, I'm not sure if you want to comment on kind of timing guidance from that point too.
Right. So we haven't given, a specific timeline for when the IND-enabling studies will provide data readouts. In the case of the mice, it's a biological system. We're breeding mice of the correct genotype, phenotype. So I don't have a firm timeline there, but all studies are on track and progressing to be completed and fit with the timing of the mid-22 IND filing. And as they complete, we will report the data.
Mm-hmm. Got it. That makes sense. And so a couple of more questions on the pig model itself. I know that pig cardiomyocytes tend to be multinucleated, whereas that's not really the case to the same extent in humans. What's the significance of that in terms of how we should interpret the transduction data from your experiments?
Yeah, great question, Geulah. And that's a really important nuance to note whenever you're looking at transduction in a pig model, is this multinucleation. So human cardiomyocytes, for context, are predominantly mononucleated. Mice, you have binucleation is common. In pig cardiomyocytes can contain up to 16 nuclei at 6 months of age. So it's a really important factor to bear in mind. So because of this, to quantify transduction, we took a pretty conservative estimate. We assume that there are eight nuclei per cardiomyocyte in this pig model, so a conservative threshold we set. And using that assumption, we quantified numbers of the proportion of vector genomes per pig cardiomyocyte with this 8 nuclei assumption. So that's the key point to note when dealing with the pig model system here.
Got it. And then you mentioned that you looked at the transduction in different tissue sections of the heart. Do you have a sense of the efficiencies within the different regions? Is it fairly evenly distributed, or are there certain regions that are more important to hit effectively for the BAG3 DCM population?
Sure. So we accepted the fact that this is a large animal heart. It would be inappropriate to sample one region and assume that every region represents that one region. So we took a very in-depth approach here. We looked at multiple rings of tissue and multiple sites across each ring, so 18 tissue sections per heart. The real core question, though, of this feasibility study was to test the hypothesis: can we just transduce the heart with this local route of administration with a low dose? And we clearly checked that box. It wasn't powered or designed to be this in-depth regional cardiac distribution study. That's partly one of the goals of our ongoing GLP toxicology study that we'll be reading out next year. But stepping back, thinking about this disease, we think that one of the most important regions to target and transduce will be the left ventricle. And it's certainly, delivering sufficient amounts of the viral gene, the BAG3 gene, to this region of the heart, we think is very important. We're certainly very encouraged by what we've seen so far.
Got it. And then in terms of the implications of these data for the clinical program in particular, how would something like AAV dosing typically translate from this setting into human, and any other color that you might have on how we should think about that for the human study?
Sure. I'll take a first pass, and I'll hand things over to Marc to add in more color too. We're taking a very thoughtful approach to scaling doses from different animal model settings, so the pig, mini pig, healthy pig setting to the human setting, and just in anticipation of our forthcoming phase 1/2 study. So we're taking a very thoughtful, considerate approach to doing so. For that, certainly in due course, we look forward to providing more color on this as we get closer to data readouts. Marc, I'll send things over to you if you want to add any more specifics on that.
Not much. Just to go back, I think, to what Maggie said about the success of gene therapy and how targeting the individual organ with your therapy seems to have led to the greatest success with the deployment of this therapy. We are going to be directly targeting the heart. We can make measurements of the heart that are fairly precise, non-invasively, with echocardiography. And we're gonna tailor our therapy both to the organ and to the patient's individual structure and functional abnormalities. We'll provide more information on that.
Got it.
We'll provide more information on that when we announce the phase 1 protocol study design.
Got it. I guess on that note, maybe we can talk a little bit more about what you have disclosed regarding the clinical plan for this program and the trial design that you've talked about thus far.
Sure. We are going to, certainly have safety as our first priority in our dosing of patients. We will start at a dose that we believe will be effective. We will enroll patients sequentially, or allowing for several weeks of observation of each individual patient in that first cohort before we move to the next patient. We are, upon completion of an initial cohort of patients, say approximately 4 or so, we will then assess what we know about the safety of the delivery of the therapy, as well as some of our non-invasive efficacy markers. We'll be looking at, cardiac structure and function, as well as patient structure, patient function, I should say, exercise capacity, functional capacity. That again, we're fortunate in cardiology, we can measure quite precisely. We'll then make a decision about moving on to a second dose as necessary and sequentially study several patients there and possibly even a third dose. At that point in time, I think that we'll have, a good idea of, what our safety profile looks like for sure. And, in addition, what our efficacy is on how patients feel and how much they can do. These are really severely limited patients, as I was speaking earlier. And, I think we'll be well able to pivot into a registrational study.
So I'd like to drill down on a couple of those points. So from your perspective in the BAG3 DCM population, what do you see as potential key relevant efficacy endpoints in this group?
Yeah, well, for example, there are patient-reported outcomes such as is obtained from the Kansas City Cardiomyopathy Questionnaire, which is an FDA recognized metric for how patients feel who have dilated cardiomyopathy. We can be quantitative as well. We can measure the amount of distance someone can walk in 6 minutes, or the 6-minute walk distance. We can use cardiopulmonary exercise testing to be even more quantitative, looking at variables such as peak oxygen consumption. How much oxygenated blood can the heart deliver to a patient's muscle when they're working out on a treadmill or an exercise bicycle? So these are several of the functional endpoints that we're going to look at, and I think that they reflect, aspects of the disease that indeed are very important to patients. Certainly longevity of life is important to patients, but many patients that I've treated who have heart failure will say that quality of life is equally, if not of greater importance to them. And I believe that regulators are recognizing this as an important outcome of any therapy, and we're going to be looking at that very carefully. I've mentioned exercise a couple of times, and the reason I mentioned that is that one of the functions of the BAG3 protein is to couple the adrenergic receptor to the calcium flow into the cell. And, when we exert ourselves, when we go for a run, for example, that is the mechanism by which the heart increases its contractility and increases blood flow to our leg muscles, for example. So I think that that's where we're going to see an early sign of a therapeutic effect of our agent. By restoring that coupling, I think that we may well see a restoration of exercise capacity in our patients, perhaps even earlier than some of the usual resting parameters that we measure in cardiology.
And I think just that for me is a really important factor for these patients. It's hopefully the group can understand we're talking about people in their late 30s, early 40s, otherwise healthy young adults going about their lives actively, potentially parents with young kids, and they're hit with a progressive form of heart failure that really reduces function tremendously and capacity. And, there are therapies that might extend life, but at what quality of life? And to be able to give back that sort of function to these patients who should have decades ahead of them is, I think, a very meaningful advantage that this therapy can provide.
Absolutely. So I wanted to touch on safety, actually. I think within the AAV space, that's been a major area of discussion, both in regards to safety signals that have been observed, for example, complement activation, myocarditis, things like that. And alongside that, there's been a lot of discussion about various immune suppression regimens that might be implemented to reduce this. So can you comment on how you guys are thinking about this and how much read across there is from, say, some of the systemic gene therapy programs that have been administered that have reported complement activation, myocarditis, and issues like that, and how that might shape the safety profile that you're expecting for this patient population?
Sure. Well, first, I'm gonna start by saying we're gonna learn from our preclinical tox studies about this. We're gonna be monitoring closely for evidence of myocarditis in our preclinical studies, and certainly monitoring for the immune response. I think that many of these adverse effects, and I'm particularly thinking of the complement-mediated ones that you mentioned, such as the thrombotic microangiopathy, are dose-related. And that's where, as Maggie mentioned earlier, and Matt presented the data about, that starting with doses that are a log to a log and a half lower than have to be given systemically, it's my hope that we'll be able to minimize these dose-related, complement-mediated adverse effects. We're certainly going to monitor our patients closely for them. We are going to immunosuppress our patients, and we're going to monitor, as I said, for signs of these. And this is where, if anything, my team's experience as transplant cardiologists is going to help our site investigators, I believe, in modulating immunosuppression as necessary.
Got it. And then in terms of the myocarditis, is there any other issues that might, is there any concerns about any specifics to the DCM population that might be relevant here in terms of the discussion?
Mm-hmm. Well, again, we're going to be very conservative. These patients do have heart failure, and I recognize that, and their cardiac reserve is less. When I think of myocarditis, and certainly with COVID-19, we've all become quite aware of the problem of viral associated myocarditis. I remember that the AV9 vector is non-replicating. So it's not, for example, perhaps as potentially dangerous as a replicating virus such as COVID-19, which is going to be an ongoing production of viral antigen. So again, we'll monitor closely. We're going to have the advantage of preemptive immunosuppression, but my hope is that it won't be too much of a problem.
Great. And then coming back to efficacy metrics, and I guess longer term clinical plan, do you have any comments on expectations around what approvable endpoints might look like in this population? And do you think a single arm trial might be feasible, or would you need to do kind of a controlled study in this type of patient group?
Well, we're actively in discussion with the regulators about, what potential endpoints might be. I think I will, bring much of what I learned from my experience developing mavacamten at MyoKardia for hypertrophic cardiomyopathy, another rare disease of young people in whom symptoms are a predominant concern, as Maggie mentioned, is true also in BAG3-associated DCM, and want to bring forward endpoints that are meaningful to patients, meaningful to clinicians, and meaningful to payers, as well as really improving patients' lives, all while showing at least evidence that we're not having any kind of detrimental effect to longevity or long-term cardiac function. We are also, again, this is a rare disease population. I would like to have the opportunity for all of our study patients to have the benefit of therapy. So we're looking into ways where, we can potentially use either, existing data or concurrently obtained population data to help inform this. And it was very nice. The FDA on Monday published an advisory document on the use of registry data and real-world evidence data on, as how to use it in a regulatory setting when seeking approval for a therapy.
Got it. So it sounds like those are options that you'll be exploring, and stay tuned to hear about how that might play out.
Yes.
Got it. That makes sense. And I think one of the other challenges within the cell and gene therapy space has been CMC issues in manufacturing. Could you touch on the strategy there and how you expect that to evolve in the coming years?
Yes, sure. A key aspect of the program for sure. So we're working with a CDMO that has a good track record in producing tox and clinical batches of AAV. They have produced high quality vector batches that have been through IND and into the clinic. So far, we're quite pleased with the yields that we're getting there. The empty full ratios are good. And the levels of impurity have also been quite good. In addition to that, we are supplementing our efforts with, I would say, ramping up our assays and analytics to keep up with where we think the regulators are heading. I think they're beginning to look more closely at assays and analytics for gene therapy CMC. So we have an experienced team on that internally that is trying to stay ahead of the curve there. So I think we're in great shape. We feel good about where we are with our materials for clinical.
Great. And so in terms of the patient population, and you mentioned earlier that the typical onset is in late 30s, early 40s. If we think about precision medicine and early genetic diagnosis expanding within the heart disease space, in theory you can envision potential earlier and earlier diagnosis, potentially even in the pediatric setting. Would there be any advantage to treating pediatric patients, or do you plan, let's say, genetic diagnosis before any kind of symptom onset, or do you plan to focus on adult patients in your clinical and potentially eventually commercial setting?
Mm-hmm. Well, I'll start. First of all, with an average age of onset in the late 30s, there are some patients who are, shall we say, are in their teens, in that 15 to 18-year-old, which is technically still considered pediatric, who do present with symptomatic heart failure. My hope is that as we become more facile with and comfortable with the safety and efficacy of our agents in either preventing or reversing some of the cardiac structural abnormalities, that we do move to identifying patients, kids with the genetic defect, and are able to treat them and to avoid the development of heart failure. I also want to bring up another point here, when you're talking about how we potentially extend this therapy, and Matt alluded to this a few moments ago. A decrease in BAG3 expression is part of a larger heart failure reduced ejection fraction phenotype. And we've seen that both clinically in explanted hearts from patients undergoing cardiac transplant not related to genetic BAG3 mutations, as well as in our usual animal models, the mice and the pigs that have had myocardial infarctions induced. So while one aspect is going to be to move to people who are genotype positive, who may not yet be expressing symptoms of heart failure, or perhaps even some of the severe structural abnormalities, another aspect is going to be, can we move to a broader heart failure population in whom decreased BAG3 expression is contributing to their cardiac function, and reverse that. And I think that both aspects are equally exciting, and looking forward to getting to work on that.
And I'll just add the BAG3. The key driver here is penetrance. It's a very penetrant disease.
Mm-hmm.
So if you're diagnosed through a cascade family screening as being a carrier, there's a very high chance you're going to develop this disease. And we're seeing as the field of genetic cardiology continues to evolve, continues to provide very supportive tailwind for us and other companies in this space. we may get to the state where treatment is warranted before onset of disease if you're a carrier. We actually already see this in one segment of DCM, another genetic segment of DCM. If you're a carrier of this disease but don't yet have symptoms, treatment is warranted. And we may get to the stage where something very similar is done for BAG3. So certainly very supportive evolution that's happening in the genetic cardiology field. And just circling back on Mark's last comment, around the extension outside of genetic BAG3 DCM, what's really common in the field, and we saw this early on, unrelated genetic heart diseases, the common red thread, lower levels of BAG3. So if you induce a heart attack in pigs, BAG3 levels drop precipitously, very similar to what we see in genetic forms of DCM. And so we've seen also in heart failure patients, our academic founder published this, patients in end-stage heart failure, again, have very, very low levels of BAG3. So that really holds the promise of if you can boost levels of BAG3, restore them, you could have a potentially an important therapeutic effect. We've already shown this, we've already published on this. In mice that have had a heart attack, we could almost completely fully restore heart function treating these mice with AAV9 BAG3. So I think it's a really exciting future ahead, not only in the field of genetic cardiology for BAG3 DCM, but also outside into these unrelated non-genetic forms of heart failure.
Great. And so on that topic, in terms of expansion to these non-genetic forms of heart failure, what does the kind of strategic decision-making look like around that? Are there specific gating factors in the BAG3 DCM program that you'd want to see certain data points to move forward, or how do you envision the decisions to advance in some of these other settings?
Sure.
Oh, go ahead, Matt.
No, I was just going to say, I think we're in a really good position. The feasibility study was an important checking of the box. It continues to support the notion that we can deliver safely low doses of vector to the heart and sufficiently transduce the heart. There's local route of administration. So that's an important gating factor, de-risking event that's happened. We've already generated, when we talk about extending BAG3 to these other patient populations, these aren't just ideas or hypotheses. These are tested. They're being published in several studies now led by our academic founders. So we have in our hands in vitro and in vivo proof of concept data already out there supporting the notion that we can, in fact, have a meaningful treatment effect in these animal model settings of acquired and more common forms of heart failure. So I think we're coming from a very, very important, promising position in terms of what we already know, the risk with discharge with our lead program and our expansion programs. And we certainly look forward to advancing these and updating you all on evolution and expansion of our pipeline next year. And Mark, Maggie, over to you if you want to add in any more color there.
No.
Yeah, no, I think that's exactly right. And I think, Geulah, you can probably hear the enthusiasm coming from both Matt and Mark as we look at the path that we're moving down at Renovacor. Both Matt and Mark come from deep backgrounds of applying genetics to cardiovascular disease. And so, their enthusiasm encourages me that we are moving in the right direction and hopefully going to really bring forth a change in the way you think about cardiovascular patients. And, what the prospects are for patients that are diagnosed with a form of heart failure at a young age. Hopefully we can change the paradigm such that it doesn't mean, years of life with a very low level of exercise capacity, functional capacity, high morbidity, in and out of the hospital. It's just really a devastating disease for the patient. And as Mark mentioned, for the families, it's a high burden on caregivers. And so when that comes on for a family with a loved one in their late 30s, early 40s, or even younger, this is a high unmet need, and we have the opportunity to really change the picture for these families.
Great. In the last few minutes, I wanted to kind of touch on some of the bigger picture questions about Renovacor in general as a company. So from your perspectives, as we head into the end of 2021 and we look at Renovacor's valuation and valuations of other companies within the space, what do you think the market is missing about Renovacor and or the broader gene therapy opportunity in heart disease?
Mm-hmm. So I think it might be exactly as somewhat of what we've been talking about today. It's new to think about gene therapy in cardiovascular disease. This has previously been more of an approach to some of the pediatric or much smaller populations. So I think the promise of a targeted approach to cardiovascular disease is one that should at some point be very exciting to the market in general, and to see what happened with companies that were looking at targeted approaches to oncology early on and how they were able to progress. I think that's where we are here today. Renovacor, we're kind of newly public, so we're wanting to get our story out there, help everybody understand what it is that we're doing. Also, one important factor I think is important for investors to understand is, while we are in the cardiovascular disease area, we are in a rare disease first. So this is a gene therapy for a rare disease indication, and our clinical program will be following a rare disease path, not the more typical large cardiovascular trials that can sometimes be more cumbersome. So important to note that, yes, it is cardiovascular, but our lead program is a rare disease group. We will be in patients with the ability to look at our efficacy endpoints that Marc mentioned in that phase 1/2 trial. So it's really exciting and transformative time, not only for the therapeutic area, but also for us as a company.
Matt, Marc, any additional thoughts on that front from your perspective?
No, I'll just say, echo Maggie and Marc's comments. It's a tremendously exciting time here. We're inspired by the potential that we have to have a meaningful impact on this really devastating disease. And we're excited by the genetics and the biology of BAG3 and the promise of extending this similar approach to other forms of heart failure. So it's a real pleasure to be part of the team and part of our mission here.
Just wanted to add in that, I think that cardiologists, particularly in heart failure, are becoming more and more aware of the need to approach segments of the population. And that, I hope investors realize that, when we talk about patient populations, that may not be the whole 6 million bucket of heart failure patients, who, as Maggie mentioned, you need, greater than 8,000 patients to show even a modest benefit. But by using precision medicine, such as we are, and targeting the mechanism of action that we can now identify with genetics and otherwise, that we can be very efficient in our drug development pathway and very efficient in really having a major impact on patients' lives. So I would, I hope that the street, as you guys call it, is going to be as aware as clinicians are of the importance of how we can use our knowledge to segment this population and bring the right drug to the right patient at the right time.
Great. So I think that brings us to the end of our hour. So I'd like to thank you all again for participating and for the great discussion that we had today.
Thank you
very much.
Thanks.